Hello friends, today we are going to discuss about one of the important topic in NEET Zoology about transgenic animals or genetically modified animals, which comes under the theme biotechnology and its application, coming under the unit biotechnology. Transgenic animals. Or genetically modified animals. What are transgenic animals or genetically modified animals? Animals whose genome or DNA has been manipulated by the possession and expression of foreign gene or genes. Simply called, an animal's genome contains foreign DNA. Why? Because for various applications, for example, increasing resistance against disease or producing biological products or drugs, etc. That means keeping view of various applications, transgenic animals has been produced. So far different transgenic animals have been produced, like transgenic rabbit, transgenic mice, transgenic cow, but 95% of transgenic animals are mice, because they had a huge role in research point of view, particularly biomedical research. Say for example, we need to test the efficacy or the safety of a vaccine. That means before applying or administering on the human beings, we use transgenic mice. Say for example, polio vaccine. The safety of the same will be subjected to different tests on the transgenic mice. So keeping in view of a different role, transgenic animals have been produced, wherein in 1974, Rudolph J. for the first time produced transgenic animal or genetically engineered, that means modified animal, say in the way of transgenic mouse, simply by introducing foreign DNA into its embryo. And for the first time, that means commercially explored transgenic animal or genetically modified animal is glowfish. Actually, glowfish is a zebrafish. Its genome contains fluorescent gene. I told you already, an animal's genome or DNA contains a particular foreign DNA or gene. Here that means the genome of zebrafish contains a fluorescent gene. What is its significance? It glows in darkness under ultraviolet radiation. Okay, transgenic animals are nothing but genome of an animal contains foreign DNA, means foreign gene. Okay, say wherein Rudolph J. in 1974 produced world's first transgenic mouse, that means transgenic animal. Whereas glowfish actually is a zebrafish, contains fluorescent gene. So it's about basic aspects of transgenic animals. Before we go into the topic, that means why we require transgenic animals, we need to understand the simplified version of the procedure to produce transgenic animals. Okay, since I told you already, transgenic animals, simply an animal's genome contains a foreign DNA, that's it. Say for example, assume I'm a genetic engineer. I need to isolate the desired gene and introduce into the host genome. That means first I have to select and isolate the desired gene, where I will get either from a cell or artificially synthesized or a genetic library. After I get the specific desired gene, I need to add certain genetic elements like promoter, terminator, and selectable markers. Okay, after that we need to introduce into the host genome. How? There are different techniques available to introduce, or that means to incorporate into the host genome, say by means of microinjection, with which directly you can inject through the nuclear envelope into the nucleus. The foreign DNA will be directly introduced into the nucleus. Or viral vectors, say retroviruses, or recently we have advanced techniques like gene targeting techniques, which form double strand breaks based on homologous recombinations. or else genome editing technologies, that means which are based on artificially engineered nucleases which form specific cuts in the DNA. Say for example, talents, transcription activator like effector nucleases. CRISPR, clustered regularly interspaced short palindromic repeats, Cas9 protein system, and zinc nucleases. So with the help of all these device techniques, we'll introduce the foreign DNA or gene into the host genome. Okay, in a simplified version, say this is the target, this is a desired gene. In simplified version I'm saying, okay, desired gene. Then with the help of techniques like like micro injection, viral vectors, or gene targeting form double strand breaks in DNA, double strand breaks based on homologous recombination, or else advanced techniques like genome editing technologies, genome editing techniques like talents, CRISPR, Cas9, zinc nucleases. With the help of all these, we'll introduce, that means this particular into the host genome, into the host genome. The desired gene will be introduced to host genome with the help of these techniques, micro injection, viral vectors, gene targeting which form double strand breaks, which is based on homologous recombination, and genome editing technologies like talents, CRISPR, Cas9, zinc nucleases. Talents means transcription activator like effector nucleases. CRISPR means clustered regularly interspaced short palindromic repeats, associated protein 9 system. Okay, with the help of these techniques, we'll introduce into the host genome. Okay, so it's about the basic procedure. Actually, in NEET point of view, keeping in view of competitive point of view, we need to explore the additional information and for our knowledge. Okay, this is a basic mechanism, but ultimately we'll introduce the foreign, and that means the desired gene, into the host genome, and we'll explore according to our need. Say for example, biomedical research. Okay, then what are the, as I discussed, why, that means why transgenic animals has been producing at a larger scale level. I told you already there are different reasons. Say for example, to produce drugs, or to increase the resistance against a particular infection. Likewise, so there are certain reasons. Say for example, to understand normal physiology and development. Then study of diseases, biological products, safety, that means vaccine safety, then chemical safety testing, these are the reasons why transgenic animals, that means most important applications are reasons why we explore transgenic animals. Normal physiology and development we'll discuss one by one. Study of diseases, then biological products, then vaccine safety, chemical safety, target testing. Say the first, normal physiology and development. Normal physiology and development. we have already discussed transgenic animals contains a foreign gene. So with this we can understand how the genes are regulated. How the genes are regulated. To understand the physiology and development, the crux point, that in the central dogma of life, is essential to understand physiology. Means, for example, a gene encodes a protein. This protein may be an enzyme or some other protein, and this enzyme regulates a biochemical function, or protein had a physiological role. That means entire physiological machinery depends on this gene level. So we need to understand how the genes are regulated. To understand this normal physiology and development, we need to understand how the genes are regulated. How the genes are regulated. This is very important. If you understand this concept, then we'd understand certain important physiological functions. Say, for example, there are complex factors responsible for growth, like insulin-like growth factors, IGFs, insulin-like growth factors. So it's a protein actually. We need to understand this insulin-like growth factor and how this protein is responsible for the growth, its biological role. For that, to understand its function, we'll introduce a gene from another species into the organism, into the transgenic animal. And this gene specifically alters the formation of IGFs. To understand its role, first we need to put some, we need to damage that function, that functional product, so that we can ascertain what are its biological effects, so that we can pull out the information regarding its biological role. If we certainly understand its abnormal effects, say that, for example, if a specific gene from a species affects its formation, like affects its structure, for example, altered its structure, then we will understand its biological role. We'll study how different biological effects are there coming out of this effect. Likewise, we will understand with respect to normal physiology and development. So with respect to different proteins, we can ascertain. So simply how genes are regulated with this concept, we can understand. Then the second aspect, study of diseases. Study of diseases. With the help of transgenic animals, we will understand how the genes are contribute to diseases. Say, for example, we deliberately cause a mutation in a gene. That means by means of a mutagen. Then we will understand how it affects the function. Say, for example, what we call transgenic models. We want to ascertain its test because we would like to know new treatments. For example, if you know these different diseases out of a mutated gene, we will investigate, we will assess regarding its treatment. So it enables us to formulate a new treatment procedure against a particular disease. So for that, we need to create a transgenic animal models. Say, for example, transgenic animal models for cancers, cystic fibrosis, rheumatoid arthritis, Alzheimer's disease. That means we can create different transgenic models for these cancer, cystic fibrosis, rheumatoid arthritis, Alzheimer's disease, so that we can investigate and come to a treatment. That means we will come to know different treatments. So for that means what we call how genes contribute to diseases. This is one of the important applications for transgenic animals. Then biological products. Biological products in the sense, say, to treat a disease we need a medicine, and this medicine contains biological products. That is biological activity, a product having an activity, but it's a costly affair. Generally the people cannot afford keeping in view of its commercial value. So with the help of transgenic animals, we can create a biological products. For example, we say, as I told you now, a foreign transgenic animal means its genome contains a foreign DNA. Say we'll introduce the foreign gene, that means into a transgenic animal, its foreign gene of our interest. Say, for example, a human protein. Say here human protein alpha 1. That means this human protein useful to treat emphysema. alpha 1 antitrypsin. Alpha 1 antitrypsin. That means this a human protein which is used to treat emphysema called alpha 1 antitrypsin. The gene responsible for this alpha 1 antitrypsin will be introduced into a transgenic animal, and the protein we can explore as much as we can. Similar to that, similar proteins we can also, the similar biological products we can also get against phenylketonuria. Phenylketonuria, or the same time cystic fibrosis. So alpha 1 antitrypsin is for emphysema, just like that phenylketonuria and cystic fibrosis. That means a biological product can be explored at the maximum level. That means for what we call economical production of biological products. Say for example, similar to that, in 1997, Rosie, the first transgenic cow has been produced, which produce alpha lactalbumin. In 1997, first transgenic cow called Rosie, it produced alpha lactalbumin. It's a human protein. That means human alpha lactalbumin in its milk at the rate of 2.4 grams per liter. 2.4 grams per liter. That means milk enriched with human protein, which is a perfect balanced diet for the human babies when compared to cow milk. That means, say we produce a transgenic cow called Rosie, and it gives milk enriched with human protein called alpha lactalbumin at the rate of 2.4 grams per liter, and this is the best nutritious milk, that means with respect to its protein content, than the cow milk. This is how its biological role. So in case of just like we have given two examples, one alpha 1 antitrypsin against emphysema, and alpha lactalbumin, that means a milk protein. Okay, then vaccine safety. I have discussed already now. Vaccine safety in the sense, say we knew that a vaccine cannot directly we cannot administer directly on the human beings. The lab tests and trials has to be done on transgenic animals. Say transgenic mice, I told you already. Transgenic mice is because out of 100%, 95% of transgenic animals are transgenic mice because of its role in biomedical research, its testing. Say before apply on the human being, in labs we test the efficacy, the safety of the vaccine on transgenic mice, so that we can understand whether it can apply on the human beings. That is, if it is successful, we can replace the monkeys as well. That means the transgenic mice can replace monkey to understand the efficacy of the vaccine. So what we call vaccine safety, this is one of the important advantage, that is application of transgenic animals. Similar to the vaccine safety, we have chemical safety testing. Otherwise called toxic or safety testing. Its procedure is similar to that of the drug toxicity testing. The same concept here. How we will do is, say we'll introduce, that means gene which is sensitive to a particular toxin. A gene which is sensitive to a particular toxin. That means we will understand how this gene becoming vulnerable or affected because of a toxin, and we will understand its biological effects. And I told you already, to understand a particular concept, a biological information or role of a particular product, we need to put some repair, we need to damage it, so that we can understand biological effect, and we can get biological information. Accordingly, we can devise new treatments. Is a whole concept. Same here also. First we will introduce a gene which is sensitive to a toxin, and we will understand its biological effects are coming out of the what we call toxin effect on the gene. say it's about chemical safety testing. Okay, so these are all different advantages. That means why we explore transgenic animals. In present class we have discussed about its basic introduction, its simplified version of procedure and applications. Okay, and in NEET point of view we need to specifically understand this biotechnological chapter. Say they might ask you an example like alpha-1 antitrypsin, which is against emphysema, and Rosie, the first transgenic cow, which is produce which protein, say like alpha lactalbumin. Likewise, they may frame questions. Okay, and for this we have referred NCERT class 12 biology textbooks and selected textbook and selected Google references. Okay, thanks for watching.